Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Basal body. 18 languages. Schematic of the eukaryotic flagellum. 1 axoneme, 2 cell. Membrane, 3 IFT, intraflagellar. Transport, 4 basal body, 5 cross. Section of flagellum, 6 triplets of microtubules of basal body. Longitudinal section through the flagellar area in Chlamydomonas reinhardtii. In the cell apex is the basal body, that is the anchoring site for a flagellum. Basal bodies originate from and. This article is about the basal body of eukaryotic flagellum. For structure at the base of bacterial flagellum, see flagellum bacterial. A basal body, synonymous with basal granule, kinetosome. And in older cytological literature with blepharoplast, is a protein structure found at the base of a eukaryotic undulipodium, cilium or flagellum. The basal body was named by Theodor Wilhelm Engelmann in 1880. 2. It is formed from a centriole and several additional protein structures, and is, essentially, a modified centriole. 4. The basal body serves as a nucleation site for the growth of the axoneme microtubules, centrioles, from which basal bodies are derived, act as anchoring sites for proteins that in turn anchor microtubules, and are known as the microtubule organizing center, MTOC. These microtubules provide structure and facilitate movement of vesicles and organelles within many eukaryotic cells. Assembly, structure, edit. Cilia and basal bodies form during quiescence or the G1 phase of the cell cycle. Before the cell enters G1 phase, i.e. before the formation of the cilium, the mother centriole serves as a component of the centrosome. In cells that are destined to have only one primary cilium, the mother centriole differentiates into the basal body upon entry into G1 or quiescence. Thus, the basal body in such a cell is derived from the centriole. The basal body differs from the mother centriole in at least 2 aspects. First, basal bodies have basal feet, which are anchored to cytoplasmic microtubules and are necessary for polarized alignment of the cilium. Second, explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Have a substructure similar to that of centrioles, with 9 peripheral microtubule triplets. See structure. At bottom center of image. Basal bodies have pinwheel-shaped transition fibers that originate from the appendages of mother centriole. 5. In multiciliated cells, however, in many cases basal bodies are not made from centrioles but are generated de novo from a special protein structure called the deuterosome. 6. Function, edit. During cell cycle dormancy, basal bodies organize primary cilia and reside at the cell cortex in proximity to plasma membrane. On cell cycle entry, cilia resorb and the basal body migrates to the nucleus where it functions to organize centrosomes. Centrioles, basal bodies, and cilia are important for mitosis, polarity, cell division, protein trafficking, signaling, motility and sensation. 7. Mutations in proteins that localize to basal bodies are associated with several human ciliary diseases, including Bardet-Biedl syndrome, 8, orofaciodigital syndrome, 9, 10, Joubert syndrome, 11, cone rod dystrophy, 12, 13, Meckel syndrome, 14, and nephronophthisis. 15. Regulation of basal body production and spatial orientation is a function of the nucleotide binding domain of tubulin. 16. Plants lack centrioles and only lower plants, such as mosses and ferns, with motile sperm have flagellar and basal bodies. 17. References, edit. 1. Engelmann, T.W. 1880. The Anatomie und Physiologie der Flimmertellen. Pflugers Arch. 23, 505. 535. 2. Bloodgood, R. 2009. From Central to Rudimentary to Primary, The History of an Underappreciated Organelle Whose Time Has Come. The Primary Cilium. Primary cilia. Methods in Cell Biology. Volume 94. pp. 352. Do. 10.1016/S0091-679X, 08, 940001-2. ISBN 9780123750242. PMID 20362083. 3. Schroeder, Jacob M. Leersen, Jesper, Komrova, Julia, Aikmanova, Anna, Thorsteinsen, Rick I. Grigoriev, Alya, Mangaso, Robert, Christensen, Sren T. Pedersen, Stin F. Geimer, Stefan, Pedersen. Lot B. 2011. EB1 and EB3 promote cilia biogenesis by several centrosome-related mechanisms. Journal of Cell Science. 124, 15 2539 2551 do. 10.1242/JCS.085852. PMC 31386999. PMID 21768326. 4. Benjamin Lewin, 2007. Cells. Jones and Bartlett Learning. p. 359. ISBN 978-0-7637-3905-8. Retrieved 28 July 2019. 5. Kim, S. Deinlacht, B.D. 2013. Assembling a Primary Cilium. Current Opinion in Cell Biology. 25. 4506511 10.1016/J.Seb.2013.04.011. PMC 37296515. PMID 237477070. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 6. Klaus Dering, Dia. Vladar, E.K. Werner, M.E. Mitchell, J.W. Huang, P. Mitchell, B.J. 2013. Deuterosome-mediated centriole biogenesis. Developmental Cell. 27, 11031112. Do. 10.1016/J.Devsel.2013.08.021. PMC 3816757. PMID 240758808. 7 Pearson, C.G. Giddings Jr., Th. Winey, M. 2009. Basal body components exhibit differential protein dynamics during nascent basal body assembly. Molecular biology of the cell. 20, 3904. 914 do. 10.1011/mbc.e08-08-08-0835. PMC 2633379. PMID 1905660. 8 Onsley, S.J. Badeno, J.L. Black, O.E. Hill, J. Hoskins, B.E. Leitch, C.C. Kim, J.C. Ross, A. J. Ikers, E.R. Tislovic, T.M. Mo, A.K. Finney, R.C. Cavender, J.C. Lewis, R. Leras, M. R. Beals, P.L. Katsanis, N. 2003. Basal body dysfunction is a likely cause of pleiotropic Bardet. Bidal syndrome. Nature. 425, 6958 628633. Bib code, 2003 nature.4256280. Do. 10.1038/nature 02030. PMID 145200415. S2 SID 4310157. 9 for anti, mi. Zullo, a. Barra, a. Beaumont, S. Mesadec, N. Studder, M. Doll, P. Franco, B. 2006. Oral facial digital type I protein is required for primary cilia formation and left-right axis. Specification. Nature Genetics. 38, 11121117 do. 10.1038/1684. PMID 163111594. S2 SID 2441702. 10 Romeo, L. Fry, A. M. Wynyard, P. J. Malcolm, S. Wolf, A. S. Feather, S. A. 2004. OFD1 is a centrosomal /basal body protein expressed during mesenchymal epithelial transition in human nephrogenesis. Journal of the American Society of Nephrology. 15, 10 2556 2568. Do. 10.1097/01.ASN.00001402220.46477.5C. PMID 154666260. S2 SID 22088755. 11 Arts, H. Doherty, D. Van Beersum, S.E. Parisi, Emma. Letboer, S.J. Gordon, N.T. Peters, T. A. Marker, T. Voisenk, K. Cartono, A. Ozuric, H. Ferran, F.M. Crows, H.Y. Wolfram, U. Brunner. H.G. Krummers, F.P. Glass, I. Noas, N.V. Ropeman, R. 2007. Mutations in the gene encoding the basal body protein UPGRIP1L, a nephrocysteine 4 interactor, cause Jalbert syndrome. Nature. Genetics. 39, 788288 do. 10.1038/2069. PMID 175584007. S2 SID 129107768. 12 carat Kobayashi, A. Higashide, T. Hamazaki, D. Kubota, S. Sakuma, H. and W. Fujimaki, T. McLaren, M.J. Welleber, R.G. Inanna, G. 2000. HRG4, UNC 119, mutation found in cone rod dystrophy causes retinal degeneration in a transgenic model. Investigative Ophthalmology and Visual Science. 41, 11. 3268 3277. PMID 11006213. 13 carat shoe, X. Fry, A. M. Tulloch, B. Manson, F. D. Crab, J. W. Kana, H. Farrer, A. J. Lennon, A. He, S. Trojan, P. Giesel, A. Wolfram, U. Vervoort, R. Swaroop, A. Wright, A. F. 2005. RPGR. ORF 15 isoform colocalizes with UP-GRIP1 at centrioles and basal bodies and interacts with nucleophosmin. Human molecular genetics. 14, 9 1183 1197 do, 10.1093/HMG/D129. PMID 15772089. 14 carat Kaitala, M. Talila, J. Salonen, R. Kopra, O. Kolskmit, N. Parvolasaki, P. Peltonen, L. Kestler, M. 2006. MKS1, encoding a component of the flagellar apparatus basal body proteome, is mutated in Meckel syndrome. Nature Genetics. 38, 2155157 do, 10.1038/1714. PMID 16415886. S2 SID 10676530. 15 carat Winkelbauer, M.E. Scaffer, J.C. Haycraft, C.J. Swoboda, P. Yoda, B.K. 2005. The C. elegans. Homologs of nephrocysteine 1 and nephrocysteine 4 are cilia transition zone proteins involved in chemosensory perception. Journal of Cell Science. 118, pt 23 5575 5587. Do. 10.1042/JCS.02665. PMID 16291722. S2 SID 16717895. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 16 carat Shang, Y. Tsao, C.C. Gorovsky, M.O. 2005. Mutational analyses reveal a novel function of the nucleotide binding domain of gamma-tubulin in the regulation of basal body biogenesis. The Journal of Cell Biology. 171, 6 1035 1044 do, 10.1083/JCB.2005081884. PMC 2171320. PMID 16344310. 17 carat Philip E. Pack, Ph.D., Cliff's Notes, AP Biology 4th edition. External links, edit. Histology image, 21804 Loa Histology Learning System at Boston University, ultrastructure of the cell, ciliated epithelium, cilia and basal bodies. Category, organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.